competitor RT-PCR. This is also one of the variant of RT-PCR. अब ये किस मकसद के लिए यूज़ किया जाता है? इसको मेनली यूज़ किया जाता है to quantify the messenger RNA or even the cDNA in the tissues. So, when the genes they are expressed, they are transcribed into messenger RNA, and this technique it can be used to quantify low amount of RNA that are produced in the tissue, as well as under in vitro conditions. This competitor RNA technique it can be used. पहले हमें ये पता होना चाहिए कि what is this competitor RNA? Competitor RNA, it is actually the synthetic RNA molecule that can be synthesized under laboratory conditions, and it can be used as a control in different PCR reactions that are based on RT-PCR. Now, what are its applications? One of the application of competitor RNA is that it can be used in diagnostic procedures. That is, when we are going to detect certain pathogens, that may be viruses, like the viruses of hepatitis or other retroviruses, whose nucleic acid is RNA. So for such diagnostic process, we can use a competitor RT-PCR. Another application is that it can be used to quantify the RNA molecule, either from certain tissues or in certain PCR reactions. So during the extraction process, this synthetic fragment of RNA, which is competitor RNA, it is added during the extraction process, and by calculating the ratio of the competitor RNA in comparison with the target RNA that we have extracted from the sample, so we can determine the losses of the target RNA. So this is also one of the application. Another application is that we can determine that whether certain inhibitors they are present during the extraction process or in the reaction mixture when we are going to conduct the PCR, because when certain RNA inhibitors are there during the extraction process or in the PCR mixture, either these sorts may be endogenous sorts of RNA inhibitors, or it may be exogenous sorts. That is, they may be present naturally in the tissue, or they may be the result of contamination from the laboratory environment. In either case, it can inhibit or it can degrade the RNA molecule. So, if RNA is there and because of such inhibitors we can't see it, we don't have any quantification. So, to eliminate such chances, one can use synthetic RNA molecule, that is competitor RNA. Because sometimes if we are going to diagnose certain pathogens like hepatitis B virus, then if RNA inhibitors are there, one of the example may be like RNAs, either exogenous source or endogenous source. So although virus is present, RNA is there, but it is degraded. So in this case, we can get certain results, and we commonly call it as false negative results, that positive result is there, but because of certain RNA inhibitors, it is degraded, it is not detected. To overcome this problem, we can also add the competitor RNA in the reaction mixture. Now, this is the endogenous source like RNAs. Some exogenous source may be the laboratory environment, like a proteinase from milk sample, heme from the blood sample, urea from the urine, or other inhibitors may be there, like if we are going to preserve the blood, so heparin, it can also be one of the inhibitor of RNA. So such all sources must be eliminated, and here competitor RNA, it can play the role. So we can again come to our main discussion that this competitor RNA, how it can be used in the quantification process. So here we now have the idea that it is laboratory synthesized fragment, competitor RNA. We can prepare the known concentrations of the competitor RNA standard, and this process you can call it as the spiking. स्पाइकिंग एक ऐसा प्रोसेस है जिसमें किसी चीज़ की जो कंसंट्रेशन है उसको शॉर्पली इंक्रीज कर दिया जाता है और इसको फिर एज ए स्टैंडर्ड यूज़ करते हैं पीसीआर रिएक्शन में. तो जब दोनों फ्रेगमेंट्स हों, टारगेट भी और हमारा कंपटीटर भी रिएक्शन मिक्सचर के अंदर एक ही वायल के अंदर, तो ये दोनों जो हैं कंपटीटर आरएनए और target RNA, they will both compete for the resources, for the primers, for other resources in the reaction mixture. So both will amplify together. And we also manage the reaction in such a way that one fragment like the competitor RNA to be larger or smaller in size as compared to the target RNA. Now what will be the advantage of the small size or the large size difference? The advantage is that कि जब हम इसको जेल पे रन करेंगे तो हम डिफ्रेंशिएट कर पाएंगे कि ये मॉलिक्यूल एक पर्टिकुलर साइज का है तो ये कंपटीटर आरएनए का है और ये जो है ये हमारा टारगेट आरएनए है. दोनों की इंटेंसिटीज को कंपेयर करके वी कैन कैलकुलेट दैट दिस इज द एक्चुअल अमाउंट ऑफ आवर टारगेट आरएनए दैट इज प्रेजेंट इन द रिएक्शन मिक्सचर. हाउ वी कैन डू इट? We will explain with the help of diagram. So this reaction, it can be performed in two steps. In step A, we can spike the RNA samples and convert to cDNA. As I told you, the spiking, the sharp increase in competitor RNA is there so that we can have different concentration or standards of the competitor RNA. So gradually the concentration of competitor DNA, it may increase. And then the cDNA, it is also there in the reaction. So two type of RNA are there. And in the next step, we can perform the amplification by using specific set of primers. Now, here in this example, the competitor RNA, it is comparatively longer in size as compared to our target RNA. So in both cases, the primer sets, they are the same. So primer recognize the same target sites and they will amplify both the competitor RNA as well as target DNA in the reaction mixture. And after amplification, then both these RNA molecules that now have been converted into cDNA, they are now separated on the agarose gel electrophoresis. Now here in this case, in the third lane, the intensity of the band is same. That indicates that they having the particular concentration in the sample, and here the concentration, it is different. As I told you that competitor DNA, it is longer in size, so it will remain behind in the gel as compared to target sequence. So finally we can conclude that RT-PCR, it is very effective technique not only to convert RNA into cDNA molecule, but it can also be used in the quantification of RNA by using competitor RNA molecule. And here the same primer is used for competitor and target RNA molecule in case if we are going to quantify this molecule.